Okay, for today's lecture, just a couple more things about the Huxley 57 model. I know you guys have all been home studying, and you probably know all of this. But actually, it's good because once we get finished Huxley 57, it's kind of all downhill. That was kind of the nasty stuff, all the math, hard to figure out. After that, it gets kind of easy. Actually, the important announcement is next Wednesday, I'm not going to be able to be here. So we're going to have to cancel class. Yeah. I know, you guys are upset. So, next Wednesday, and I've been doing this 50 years, and this is unfortunately the first time I've had a conflict that I really couldn't resolve. What we're going to do is Monday, I will pick up with a few more things. But this test will really only cover the 3 lectures I gave. The Monday stuff should be good. You'll learn a lot. Hopefully, it's usually the most fun lecture. And then Monday after class, I'll have office hours from 5 to 7. We'll get a room in Clark. That's generally fun too. So we can go over old tests, homework problems, whatever you want to do. Usually people come. It's a good chance for me to get to meet people. So I enjoy it, and I think the students do too. I also should say that we have internships at IBM. If people are interested, email me about New Year's time and see. And we also have postdoc positions. Anyway, let's go on. Okay, so as I say, Huxley 57 is the worst part of my section. So you've gotten pretty much through that, but just a couple more slides. Okay, part of how we motivated Huxley 57 was that this classical non-parameter model fails in that if you look at the force-velocity relationship, it would be a straight line. But the real one has this curvilinear nature, it's complicated, and it's hard to understand where it comes from, and that really drove the Huxley 57 model. But at some level, you go back, I mean, as I was saying, people still use this as a very rough model of muscle, and at some level, even the real muscle, even though it's curvilinear, looks a bit like a viscosity element. So where is this viscosity nature coming from? And really what it comes from is the way that the cross bridges redistribute as you try to move the muscle in positive or negative velocities. So this is at 0 velocity or fixed length. And then as you move to faster shortening velocities, this profile changes as we discussed. And the net effect of that really does sort of give it a viscosity kind of effect. As it tries to move faster, you get more draggers out here to slow it down. So at some level, there is this sort of viscosity effect. And it just comes from the way that cross bridges redistribute as you move the muscle, and it takes on a non-zero velocity. And then we didn't get to talk about it, and because our time is short, we won't get into negative velocities with Huxley model. But in the homework, you'll get to try out negative velocities, and then what you'll see is in the negative velocities, you also get this kind of viscosity effect. Now, someone brought up, I think last time, about why is the cross bridge want to reach out here and attach? In the Huxley model, there's an F that increases as you go out towards H. And so it's saying, now that we understand what myosin does, really probably what we can think of is the myosin reaches up more or less in the equilibrium position, and the head attaches, and then it makes this quick rotation. And when it puts the rotation, it effectively puts that strain in the tail region. And so even though, in Huxley model, it reaches out there and attaches here, it attaches more or less at 0 and rotates quickly. So we won't get to do the 4th lecture in much detail, but when you begin to build models, what you see is that very often some states you may know exist, but if they're very quick, they're not very highly populated. So it's sort of like you can ignore them. And so, you know, as we were saying, all models, you know, no model is right in this, it fully captures reality. So sometimes we can, you know, perhaps ignore a state or merge two states together and make a composite state. So this attachment out here with the positive distortion, you can think of really as a composite of two states attaching and then the head quickly rotating. Now, I don't think we really got to talk about it much, but when cross-bridge is attached, there's this kind of weakly bound, and I think I just say electrostatic. So really detached often is called weakly bound because whether it's actually detached or just sort of weakly attached to the actin, it's sort of like the detached state. And then it finds this preferred binding conformation, and then it becomes strongly bound. And after it becomes strongly bound, then the head can rotate. And, you know, so here, this was one of the slides from the first day. We started here in the Riger cross-bridge position. You add ATP, now the cross-bridge can detach. As I say, detached actually, maybe it doesn't mean just floating around out there. It may just be sliding along the actin, but not really strongly attached. You cleave the ATP, and somehow you store the energy. And at some point, which would be about here, is where it becomes strongly attached. And then once it becomes strongly attached, now the head can rotate. And this is called the power stroke. So when you have a power stroke, you put this delta X in of more strain. So again, so what does delta X relate to in the Huxley model? Well, delta X is approximately H, how far out a cross bridge can reach and attach. So that value is sort of like the delta X again. Sort of attaches near zero strain, and then the head rotates, putting that delta X amount of strain in the cross bridge. So I talked about this briefly, but now it's time you should think about it again, which is, this is really the governing partial differential equation. Here, which says its partial n with respect to t is equal to minus the velocity times the partial n versus x. So it's sort of like there's this balance between what's going on with respect to time and what's going on with respect to distance. So you sort of think about motion effects versus attachment effects or attachment events. So I was saying, you had this kind of profile here. If you had a cross bridge attached at, say, 0.5, it really could have gotten there by one of two ways. It could have attached at 0.5, or it could have attached somewhere larger, and then it got pulled down to 0.5. So at any given position, you can think of, you know, cross bridges either get there because of attaching there, or attaching somewhere larger and getting pulled there. If we had a negative velocity to go the other way, you would attach, say, a lower distortion and get pulled out. So it's really a balance of what's, and actually to be complete, it's really what's attaching and detaching at a given distortion, which is these terms, times, is being sort of in balance with what is sliding to that particular distortion. Okay. We're almost there. We're almost through the Huxley 57. The last part here is there's this Excel-based simulation that David Yu and I put together back around the turn of the century, but it's just as fun today. So basically, what's going to go on in this simulation is that in the original Huxley 57 model that we just derived, we derived for different distortion values. And here what we're going to do is we're going to split it up in discrete steps. So this is like zero distortion, and each of these discrete steps is a nanometer, so we got the plus 15 or minus 15. And then now we're going to split time into discrete steps. So in the original derivation, everything was continuous space or continuous time. Now what we're going to do is solve it on a discrete grid. So we break up, again, space into discrete steps and time into discrete steps. And then we can say implement these attachment and detachment rates. These were originally defined like this, which are continuous. Because we're only working at certain distortion values, now we can define them at discrete points. So this kind of representation really just mirrors that. And then, luckily, to run this model, it's super simple, right? Velocity is a parameter in Huxley model. You plug in a velocity, and you read out the force. That's all you have to do. So it's very simple. But what in the homework, what I'm going to do is make you look at the graphs and try to interpret what's going on and get some insight about, how things function. So, because we're short on time, I'm not going to go through this. If you're interested, if you want to understand how to construct models, this is going to show you how we, again, turn this continuous time system into a discrete time system. Jumping ahead, now, we know now what happens when it's at zero velocity, where you have this high uniform value for attachment. So n is this value of about 0.8. And 0 for greater than 8, and 0 for negative values. So in this model, what we're going to do is, instead of like the analytic derivation that we did, where we assumed it was a steady state, here what we do is we actually start at an arbitrary state and evolve the system forward in time until we reach steady state. So this is a typical thing you do in simulations. It's very often, in very complicated systems, it's hard to figure out what is the steady state analytical solution. So you just start it in some arbitrary state, you run the thing forwards, and eventually you hit steady state. And that's the way that this is implemented. These are different model results for higher velocities. This is for grams. So this is the point here. If we look at this, then this is over time. So we start everything around 0, and then it comes up to some value, and at some long amount of time, approximating t going to infinity, you can pick off the values, and that's the way this works. You'll get to see it in the homework. So again, these were the continuous time representations. In the derivation, we had this W hat, which was this probability density function. We also talked about the attachment of a cross bridge generates this much force, which is k times x. So this would be the plot of force for attached cross bridge. And in this model, we actually stick these two together, and this is going to be kx times w hat. Actually, I think, I'm sorry, it's right in this slide. It's wrong in the next slide. But you put these two things together, just multiply them together, and then you get this in this discrete time space. And then, so this is the n value. This is kx times w hat. And then you multiply these together, and now this function is something that we actually didn't plot in the last lecture. I think your notes say h hat here. It should be w hat. Again, we used to use h hat. But here is, we're talking about taking the expected value. So this is the signal where if you're at, where you can actually take the area under this curve and the area above this curve. If it's at maximum velocity, then they're equal. So this contains both the contribution of probability of attachment, also the contribution that is the more extended the cross bridge, the more force it produces. So you'll get to see this in the homework. Okay, so what we're going to do now is, okay, Huxley model is finished for now. So relax, it gets much easier. Sit back and enjoy this part of it. Okay, calcium regulation. So for the most part, really we've talked about muscle being fully on. We have talked about twitches, but we didn't explore it much. So now we have to talk about how does muscle turn on and off. So in striated muscle, which is cardiac and skeletal, it works like this. You have a thin filament, which is this two-stranded helix of actin, and on it you have troponin, which is represented here by this ball, and then you also have something called tropomyosin. And these two work together, and in low calcium they tend to block these binding sites. If you add calcium, there's a shift in the position, exposing binding sites, and now a cross bridge can get in and bind. So this is the basic scheme of it here. So this is what you're going to see in the textbook. Let's look at the different proteins. So tropomyosin is sort of this rod-like protein, and it sits in the groove of this two-stranded helix of actin, and it spans about 7 actin monomers. So the interesting thing here is it connects end to end. So it's sort of like it wraps around the thin filament. You also have a protein complex called troponin. There's actually 3 different subunits. Troponin C is C because it binds calcium. It's similar to a thing called calmodulin, which is this dumbbell-shaped protein that binds 4 calcium ionos. So it's similar to that. This is dumbbell-shaped. And what happens is you have these 2 high-affinity sites. So down here you have calcium bound all the time, and it doesn't do much. It just sits there. The calcium binding to the other end is what regulates muscle contraction. So there's different kinds of troponin. In cardiac muscle, there's one calcium that binds here that's regulatory. The other binding site doesn't work right. That's in cardiac and slow twitch skeletal muscle. In fast twitch skeletal muscle, there's 2 binding sites. So there's 2 sites that are regulatory. Now troponin I is another subunit. We're going to show you in a moment what it does, but it has an inhibitory role. It switches between tight binding to actin and then tight binding to troponin. So again, we'll show that in a second. It's also a site of regulation by a number of second messengers, which we're not going to get into it. But there is some regulation in the system by other features besides calcium. And then there's troponin T, which is kind of its role as a linker. So this is troponin I because it's inhibitory, troponin T, and actually I don't know what the T stands for. So a low calcium. Have you guys heard the term diastole yet? So, okay, so you're going to have all the cardiac lectures coming. So this is like the rest state in the heart, diastole, and then the systole is contracted state. So every heartbeat, you go from resting, diastole, to systole when the heart beats. So that's just labeled here. What's important here is this is low calcium concentration, and this is high calcium concentration. So at low calcium concentration, we tend not to have calcium-bound troponin C, and then troponin I, which is this other dumbbell-shaped protein, really sits mainly connected to actin. So maybe I should spell these out. This is troponin C here. This is troponin I. This is troponin T, which is kind of like a linker again, which links troponin complex to tropomyosin. And the tropomyosin spans 7 actins and it goes end to end. So this is one set of tropomyosin, then here's another tropomyosin. So when we bind calcium up to this lobe of troponin C, then troponin I shifts up, and now it moves troponin T and tropomyosin and exposes the binding sites to bind myosin. So this is another view of it. This is electron micrograph reconstruction. And so this is showing in the low calcium, the tropomyosin is sitting here, which blocks the myosin binding sites. And then you add calcium, and now it shifts, and then it's shown in red of the myosin binding sites. Now it says, what should it look like for intermediate levels of calcium? So if this is low and this is high, what happens in the middle? And unfortunately, we're not really going to get to that problem. We'll get to a little bit in the last lecture. So this is a cross-sectional view. So now if you cut the thin filament and you're looking down, axially down the thin filament. So again, thin filament is a two-stranded helix of actin. So this is where the proteins sit on the outside. This TM is tropomyosin, troponin T, troponin I. This P here is the site where it can be regulated. Here's troponin C. So this is low calcium here. And then what it's doing is, you can see where it's blocking this myosin from coming in. As I say, myosin actually kind of wraps around the actin. So you can see a little better view in this. So this is a myosin head here. We add calcium, then this whole structure sort of rotates out. And it's going to shift the tropomyosin to this position. So now tropomyosin is shifted out, and now myosin combined. Now, this shows something interesting. So this is low calcium tropomyosin state. Here is high calcium. And then it shifts even more when you bind the cross bridge. That's what this thing says. So this is low calcium, high calcium, plus S1. S1 refers to a myosin head. S1 is another name for a myosin head. So that shows what happens. So here's an animation done by my collaborator, Peter DeToon. So this labels all the pieces. So calcium, this is one. Okay. So this is very interesting line, but it's not moving. Okay. So we're not going to get to see that. But I'll tell you what you would have seen if it worked. So basically, what happens is calcium comes bouncing along and it binds here to troponin C. This green protein, which is troponin I, rotates out. And then it shifts the troponin T and tropomyosin so this cross bridge can get in and bind. If you download this, this probably will work on your laptop. I think it's one of these video card problems. And usually, well, too, since we're a little short on time, what I can show you is that, just go back a few slides. One of the things that happens is that you get some shift here from binding calcium, but when cross bridges come in to bind, it actually pushes these proteins a little more further. So it's kind of like the system begins to turn on with calcium, and then the addition of cross bridges turn it on further. And in fact, what happens is these strongly bound cross bridges will hold the system on, even after calcium is associated. So the point here really is that if you look at this from your standard textbook kind of description, you go, oh, well, this is very simple. Calcium is a switch. It turns the system on. You get cross bridges. And then you take lower calcium. It's like you flip the light switch off, and the thing turns off. But really what happens is very interactive. So the presence of cross bridges themselves can keep the system on. And that has ramifications for the dynamics. Again, I'm trying to get people out of the standard kind of textbook understanding about how things work, something that's a little more mechanistic and closer to real life. So I'm telling you what, since you can read these, and I'm going to do nothing but read them to you, I'm going to skip ahead and read these. By the way, the homeworks, I should say, the homeworks, I'll give out also some practice test questions. The homework, the test will be just like the homeworks, almost exactly. Sometimes almost the same questions. I mean, there's going to be no surprises here. So you're going to have to solve one of these classical one-parameter models. I'm going to make you do a bunch of things with the Huxley model. And then pretty much there's going to be something about sarcomere overlap and I bands and A bands. And then I'm going to ask you a bunch of questions about sort of different aspects of these different muscle types and the proteins, like how many. I might say, what are the subunits of troponin, troponin C, troponin I, troponin T. I make it simple because it's always true-false, so you got a 50-50 chance of getting it right, even if you don't study. So. Okay, so I put these slides in just last year because when I learned about muscle, there was thick and thin filaments, or troponin and tropomyosin. That was sort of all that was known. Little by little, we know more about the system. There's this other thing called myosin-binding protein C. They used to just call it C protein, now its preferred name is myosin-binding protein C. And this is a protein which sits here in the sarcomere. So remember, the A band was the thick filaments. So it sits here between, sort of on the thick filament. And it's a complicated protein. It really bridges the thick and thin filaments, which exists in the C zone. So we can call the C zone here, where these myosin binding protein C is. And it's a large protein that connects actin, myosin, and titin. And it helps to form the sarcomere. So if you get rid of this protein, you either genetically modify the animal or you get rid of it some other way, what you find is that the sarcomere is formed. They're just a little malformed. And so it's kind of one of these, unfortunately complex things in biology where it's important for the system, but it's not absolutely essential. So probably it's something that evolved later after the actin and the myosin. The sarcomere was set up. This came later and seems to have a regulatory role. So there's different isoforms for cardiac, fast skeletal, slow skeletal muscles. So there's slightly different isoforms. Don't worry about knowing all of these different domains. I won't ask you anything at that level of granularity. The only piece that's really important is that the cardiac one has this place that can be phosphorylated. And so that's a place where the system can be modulated. So people here may not have had the action of kinases and phosphatases and how proteins get phosphorylated. So here's a very, very quick lesson here. So there's a very common motif in cellular biology. You see this all over the place. This is called a second messenger system. So one of these is that you have a kinase, and what happens is it will take this phosphate group off of ATP and stick it on a protein, on a serine, or I think it's, I can't remember which one it goes, but there's a couple of different residues where you can stick on this phosphate group. So kinase puts on a target protein, and once this happens, it changes the function of that protein. So what was the native or APO1 now becomes changed when you stick on a phosphate group, change its function. And then there's a phosphatase here which will then strip it back off and turn the target protein back into its other shape and its other function. It changes the function. So these things come in pair, a kinase and a phosphatase. As engineers, you should always think if you turn something off, it's not valuable. If you can turn something on, it's not valuable unless you can turn it off. So these two work in antagonistic pairs. So what's going on with myosin binding protein C? So this is showing it in its dephosphorylated state here. And it sort of connects here to thick filament with myosin. Remember, this is myosin tails and titin. So it connects down here. It travels up along the myosin. And then it also attaches here to actin. Now this is complex, right? You can't see these proteins in action. You can only try to really infer what they're doing. And again, this is a really hard system because you can't just take it apart into pieces and see what each piece does. It really, when it's in this complex, structure is where the functions, the true functions lie. But here it appears that it's going to bind both to the thick filament and to the thin filament, and somehow it puts a brake onto that actin and myosin interactions. So when you phosphorylate this protein here in this end domain, what happens is it releases the brake. It allows the cross bridges to cycle, more attachments, and increases the amount of force. And it also increases the calcium sensitivity. Again, even though calcium is a switch that turns on and off the system, myosin itself is very interactive, and so more cross bridges actually changes the calcium sensitivity. If we get that far into the next lecture, I'll explain that a little more. This is just a picture here, an animation. So what this is showing is the normal protein. So when it's like this, it's putting a brake on the myosin. When you phosphorylate it, this area can pop out and makes the head sort of able to move more, increasing the actin-myosin reactions. This is one here where you genetically modified the animal, and now you truncated this protein, so it's missing the end domain, and so effectively it gets rid of the effect on the myosin head. This is just a conceptual model. Again, we don't really understand exactly how these things work and their function, but the point here is that, you know, this is part of what we know about muscle, and to try to be complete about what I teach you. I need to teach you about myosin binding protein C. So it has a lot of roles. It seems to have a structural role in that it helps make uniform sarcomeres. It has a regulatory role. It modulates calcium sensitivity and contraction force in cardiac muscle. And so neither of these is essential. If you get rid of this protein and knock out mice, the thing will live, but it may end up in hypertrophy, which is enlarged heart or heart failure. The problem with this is any kind of insult to the heart can really just phenotype of hypertrophy and heart failure. This is coming in later lectures, you'll hear about this. But probably this point is important, is to suggest that sarcomere and contractile proteins have some kind of tuning and optimization. And this particular protein is something that tunes the system and optimizes it. Great, so now we get to go on to different muscle types. So skeletal muscles. Skeletal muscles are the ones that are on our skeleton that move our muscles. I'm going to move our limbs, keep us upright. So skeletal muscles run from tendon to tendon. So say your bicep muscle, the cell actually goes from one tendon down to another. It's a big, more nucleated cell. So if we went down from the full muscle to the deltoid, then you go down, you see the fasciculus, and then you can go down to another level of muscle fiber, and then below muscle fiber is a myofibril. When you get down to myofibril, you can actually now see the sarcomeres. Remember, the sarcomere is not flat, it's strong, but actually a three-dimensional structure. So these are different levels of organization. This slide, I'm not really going to ask you about unit 10A versus 510A. The point here really is just that muscles structure is kind of determined by what they have to do. So in this muscle here, which has a strap muscle, basically you have a lot of sarcomeres in series. So what this is going to do in a way is optimize for contraction velocity. So if you look at the velocity of one end versus the other. So even though each sarcomere may contract at the same rate, which they do, when you stack a bunch of them on top of each other, then end to end, you're going to get a faster contraction. Now other muscles are set up so you have more sarcomeres in parallel. And when you put things in parallel, the shortening velocity is the same, but the force goes up. So this is just showing really that there are different kinds of muscles. And for instance, when you get here, you're going to put more sarcomeres in, even though now force transmitted is a slightly different angle. You may do that just so you can put in more sarcomeres in parallel. This is showing another example. So gastrocnemius here has the fibers going in this direction, and that allows you to generate more force. Sartorius, this is more like the strap muscle where you have more things in parallel, and so, more things in series. So again, if it's this structure where you have more sarcomeres in parallel, then this, you get more force. This ensemble would have 3 times the force of a single sarcomere, but it would have the same shortening velocity. When you put things in series like this, now the force of this ensemble is the same as one sarcomere, but the end-to-end shortening velocity is going up if you add sarcomeres. So these two would have different force-velocity relationships for the whole muscle. Now again, if you get down to the sarcomere level, that's different. I mean, the sarcomeres are sort of the basic unit. They would have the same force-velocity relationship, but depending on how you arrange the sarcomeres, so you can get different behaviors. So this was from the first lecture. Just recall that there's this force-length relationship. This is showing here the total tension as you go to a longer length. Hard to see here, but this corresponds to sarcomere length. You have an ascending limb, a plateau, and a descending limb in skeletal muscle. If you then if you don't activate the muscle, you can also measure a passive force. And so, where's the passive force coming from? So there's tighten. So just like we talked about tighten in cardiac muscle, there's also tighten in skeletal muscle. But it's set up so it operates at a somewhat longer length. So in cardiac muscle, recall that there's really, it's fixed, so it can't go past about 2.3 or about 2.4 centimeters. So that's all you can really stretch a cardiac. So skeletal muscle you can stretch much further. So that's why the passive force curve is somewhat different. So if we go back to our classical lump parameter models, and we talk about this parallel elastic element. Well, what is the parallel elastic element? Part of it is tightened. So as we stretch the muscle, it pulls back as part of this parallel elastic element. There's also collagen that surrounds the muscle. So this is the connective tissue, and that acts also like a parallel elastic element. So hopefully this is going to tie it together a little bit. Some of the early lectures we had this model based on these simple elements, and then what does it correspond to in the real muscle? How about the series elastic element? Does anyone have a sense what that corresponds to? So what it is you have ligaments at the end of the muscle, which is somewhat rubbery and compliant. So that gives you some of your, could be part of the series elastic element. But if you have cardiac muscle doesn't have ligaments at the end. So there, you grab the muscle with something, and if you damage it, and that part stops being contracting, and now becomes kind of compliant. So sometimes it's just sort of the damaged end of how you attach the muscle that gives you that series elastic element. This was another picture from before. Just the point here is that there's really a lot of collagen surrounding the skeletal muscle. And so again, this sits on the outside, provides some of the series, the parallel elastic element effect. So how do we activate skeletal muscle? So here you can see these are myofibrils. So in a way, the whole muscle fiber is one big cell, and it's full of these myofibrils. And there's a sarcolemma or a cell surface around it. And then the important thing about striated muscle, the skeletal and cardiac, is they have transverse tubules. Actually, I'm sorry. All the skeletal muscle has these transverse tubules. There are actually some cardiac muscle that doesn't. But transverse tubule or T-tubule is sort of like this invagination that runs deep into the muscle. And so that allows the action potential on the cell surface to pass down into the interior of the muscle. And why do you want that? Well, what you want that is, why you want that is because calcium is released from this internal store called sarcoplasmic reticulum. So, sarcoplasmic reticulum. So, it's kind of like a sort of cistern. It's sort of a reservoir of calcium that exists inside of muscle cells that can be quickly released. So if you look at something like a frog or an amphibian, it doesn't really have much sarcoplasmic reticulum. What this seems to be is an adaptation that allows the muscle to respond more quickly. So you see this in warm-blooded creatures. So you can imagine that you could have calcium, when you wanted to activate the muscle, one thing you could do is have calcium come from outside of the cell inside. And that does happen, but there's also the secondary system where you get secondary release out of sarcoplasmic reticulum. Now it's sort of set up like this. The transverse tubules exist near the Z lines. So here's a Z line, here's another Z line. And calcium gets released from here, it goes out, and then it gets taken back in. So it gets released, something called the junctional SR or JSR, and then it gets taken back up in the network SR or NSR. This is in the next slide. Oh, no, I'm sorry, it's not in the next slide. But that's the basic idea. So skeletal muscles are activated by a motor neuron. It comes, so this is a nerve coming in and it terminates on the muscle. It has a neurotransmitter, acetylcholine, which after this gets activated, it will activate the sarcolemma. It'll give you an action potential, which is going to travel along the length of the muscle, and then it dives down to these T tubules. So as this becomes depolarized, calcium is led into these voltage-gated calcium channels. And in skeletal muscle, they connect with something called a ryanodine receptor, which is on sarcoplasmic reticulum. So it's action potential comes down, lets calcium in, and then you get a secondary release of calcium out of sarcoplasmic reticulum. Now the critical point here is in skeletal muscle, there's this foot process that connects this channel to this channel. And basically when the voltage changes here, it causes calcium release. So this is sort of voltage-induced calcium release. Now, after calcium comes out of the sarcoplasmic reticulum, it can go out and activate the actin-myosin, but then for relaxation to occur, the calcium is either pumped back into the sarcoplasmic reticulum through this, which is called a circopump, or else it's pumped out across the cell membrane. So there's two ways it goes out across the cell membrane. One is the sodium-calcium exchanger, and this works by letting sodium come into the cell, and when sodium comes in, calcium goes out. So it's the exchanger. There's also this, which is called a sarcolemmal calcium pump, and this uses the energy of ATP to extrude calcium across the cell membrane. So there's two ways that calcium gets in and out across the cell membrane. So this is going to be the second laser pointer for David Drive. Okay. Thought I brought another one here. Okay, sorry about that. Okay, so this is another view of this. So muscle cells are excitable like neurons. And so when you learn about neurons, you can sort of excite them, you can get a blip in the voltage. When the voltage gets up to a threshold level, you get an action potential, and it's all or nothing. That is a neuron. So once you get an action potential, then essentially all the events, ensuing events happen, and that is action potential goes down to the tubule, you get a change in voltage, there's voltage-gated calcium channels that come in, then calcium comes into the cell, then you also have this secondary release from sarcoplasmic reticulum. This view nicely shows how this should really line up with the Z line, but calcium comes out near the Z lines, it floods into the sarcomere activates it, and then the calcium gets pumped back into sarcoplasmic reticulum. So this is junctional SR, and then this is network SR. So calcium goes back in. So if you have warm-blooded creatures, most of the calcium is cycling out of sarcoplasmic reticulum, and less of it goes in and out across the cell membrane. If you have like a frog, it would be more about the cell membrane mechanisms. So the skeletal muscle, when we activate it, so we can get single twitches. And the skeletal muscle, these twitches are going to be about the same. So it's kind of like the muscle is a bit more all or none, like a neuron. You know, in principle, neurons are, every spike is the same. Well, every twitch is more or less the same here in skeletal muscle. Now what happens is, you know, if you increase the frequency, you get tetanus and it builds up. Now what I'm going to say is probably most of that buildup happens because of the slowness in the cross bridges attaching and detaching. And probably events at the level of calcium cycling are more all or nothing. And so this is a somewhat old slide. But what this is showing is that, look at the A case here, is that calcium will go up fairly quickly, and then it takes much longer for, so this is the voltage, this is the calcium, and then this is the force. And so what happens is that calcium goes up early, and it's coming down while force is still growing. So the main point here is the calcium mechanisms tend to be fast, but the attachment and detachment of cross bridges tend to be slower. And this is shown here. So if you look at the activation, which is shown in the purple here, that's like a neuron, very short duration, just a few milliseconds. And then if you look at twitch force, it occurs much later. So this first one, ocular muscle, is really about the fastest. It's going to be one of the fastest muscles you can find. But in this muscle, the electrical event is over and done before you get any force. So you get first electrical, then calcium, and then force. And you can see here. So this is a fast muscle. You can also get these other muscles like gastrocnemius or soleus. This one's fast twitch, so it's type 2. And then you can also have slow twitch, which is type 1. So this gives you an idea of the temporal differences between the fast twitch versus the slow twitch. And then this is the setup for skeletal muscle. So what happens is you have a signal state to move one of your muscles. It comes from your brain down your spinal column, say if it's going to say your leg. And the motor neuron actually sits in the spine. And so there's a motor neuron, and then you have an axon that goes from that motor neuron all the way out to your muscle. So, you know, if it's something, say, something in your foot, action motor neurons in your spine, you have a long axon that will travel all the way down to where the muscle is. There's two kinds of, so you could add something else. So basically, once this activates, then this is a very secure system. So you activate the motor neuron, and then you get an activation of the muscle, and you get a twitch. So basically, whatever's happening here gets mirrored down at the muscle. Now, there's two kinds of muscle units that we're going to discuss here. There's a large motor unit and a small motor unit. So this is larger, fast conducting, relatively inexcitable. Small one, small, slow conducting, relatively excitable. So those are for the axons. For the muscle fibers themselves, the large ones are these type 2, which are fast twitch. And large, glycolytic, which means that they have stored energy that runs out. So the way you can think about it is these guys are the sprinters, these guys are the long distance runners. So these are more oxidative. They can continue to go because they have oxidative phosphorylation. They're making enough ATP, keeping cells going. And so sort of functionally what happens here is that when you need to contract your muscle, you tend to use these first, And then if you really need sort of maximal activation, maximum force, maximum speed, then you recruit these later. And so if we think about skeletal muscle, then I think the important take-home message and the thing that may show up on your test is what are the ways that we can modulate force in our skeletal muscles? So one way we can do it is by choosing which kind of fibers we choose. We can choose large ones or small ones. We can also pick which fibers we activate. So typically you have a lot of fibers together in the muscle, and your brain is figuring out how to activate those in different sequences to give you the amount of force. Now skeletal muscle, you also have this ability to tetanize. So if you activate very quickly, you can sort of tetanize the muscle and get more force. So the skeletal muscle, you can have the activation pattern, whether it's single twitches or tetanus. You can change the number of fibers recruited, and you can also change which type of fibers are recruited. So that's really this point, the last part, which is you can choose the number of units recruited to match a given task. Okay, now we're going to go on the cardiac muscle. Cardiac muscle is for your heart. So this is a big cross-section of the heart. Got 4 ventricles. Four chambers, 2 atria, and 2 ventricles. And then basically it activates from the sinoatrial node, which is going to start action potential propagating to the atria, and then there's specialized conducting fibers, which then take it down into the ventricles. And the point of this really is that unlike skeletal muscle where you can recruit different fibers, in the heart you get every single muscle cell in every heartbeat. So basically your whole life, from being child to adult, every heartbeat you get every muscle cell. And only in pathological conditions is that going to change, and if you're in that condition, it's bad news. So basically you get every cell, so you can't recruit more or fewer heart cells to modulate the force of contraction. So this is going to be one of the themes, so we have to think about how do we modulate force of contraction in the heart. So this shows the microstructure of it. So cardiac cells, sometimes called cardiomyocytes, so they're also excitable cells. In this case, the action potential is passed cell to cell. So this is probably because of what the heart has to do to effectively contract and inject blood. It works better if there's a slower propagation of electrical signal. So again, things, electrical signals pass cell to cell. Let's just look at a few things here. Over on this side, these are sarcomeres. So this is what you know with Z lines and the A bands and the I bands. So these are sarcomeres here. So you see the same things here. This also has a red blood cell and a capillary. So you need a lot of oxygen to run the heart, a lot of blood vessels, as is shown. And this, you also have a lot of mitochondria. So you need to generate a lot of ATP. So you can see mitochondria here. And then you can see sarcoplasmic reticulum. Again, this is reservoir of calcium. Now the important things here in heart is that you have gap junctions. So the gap junctions are little proteins that allow ions to pass between the cells and allows the action potential to pass from one cell to another. There's something called the intercalated disc or intercalated disc, which is the connection between the cells. There's also connective tissue inside the heart. Again, there's the polyune outside the heart cells that we talked about, which generates some of the passive force. So within the intercalated disc, for mechanical connections, there's fascia adherens and macula adherens, which are in the next slide. So fascia adherens are anchoring sites for actin, and they mechanically connect to the closest sarcomere across the cell. So here is FA. So this is the fascia adherens. So basically what's going to happen is you're going to butt the sarcomeres of one cell up to the sarcomeres of the next cell, right? I mean, you couldn't have these cells disconnecting, or you couldn't transmit force from cell to cell, which is what you're going to need to have coordinated contraction. So these fascia adherens attach cell to cell. You have also macula adherens, which bind intermediate fibers, also known as desmin, which is part of the cytoskeleton. And it's another way cells are connected together. So within the cell, there's three main components in the cytoskeleton. So I don't know how much biology we have, but inside the cell's not just a bag of water, there's a cytoskeleton. And so actin's an important component. This isn't any cell is going to have actin in it. Now, in muscle cells, we know actin is a thin filament, but there is actin in cells. There's also these intermediate filaments, also known as desmin, and then there's microtubules. So those are the basic components. So there is intermediate filaments in cardiac cells, and they're connected together as these. It's also called a desmosome. So this is a picture of what happens in cardiac cells. So probably should have said it in the skeletal part. So we call it excitation contraction coupling. And so excitation is when you excite the cell with an action potential, and then somehow that gets translated into contraction, and then the intermediate. That whole sequence is often called excitation contraction coupling. This was a picture developed by Donald Burrs. He wrote a book on excitation contraction coupling. And this picture is how everybody draws it. Although in reality, this is perhaps not so close to a real cell. And you notice that in the picture of a cardiac cell, it's packed full of sarcomeres. I mean, this is just one teeny sarcomere. It's because this guy's really focused on these proteins, so it's sort of his world. And the monofilament isn't really his thing. But in reality, the cell is mainly full of these sarcomeres. But let's look at what happens. So when the cell excites, you get calcium coming in these voltage-gated calcium channels. Now the important thing in cardiac cells is the calcium comes in and it triggers a secondary release of calcium from sarcoplasmic reticulum. And in this case, it's not from the voltage itself, it's actually coupled by the calcium. So this is called calcium-induced calcium release. Calcium comes in and causes a secondary release of calcium. So calcium, some of the calcium coming in comes from the sarcolemmal channels themselves if you go to activate the monofilaments, but most of it comes from sarcoplasmic reticulum. Now there's a sodium-calcium exchanger, and so sort of interestingly, what this does is it extrudes calcium, it extrudes calcium by letting in sodium, but it's voltage sensitive. So actually early in the action potential, it lets calcium in, which is shown here. So these transporters or exchangers really run off an electrochemical gradient, and when you change the voltage, sometimes they flip directions. And in the heart, that actually happens. Initially, this exchanger brings calcium in, and then later during relaxation, it also takes calcium out. So this is a bit of a complicated diagram. So let's go through it one more time. So calcium comes into the cell, either it can be voltage-gated calcium channels, it can come into the cell, directly activate the monofilaments. Most of the calcium comes in and triggers a secondary release, this calcium-induced calcium release. Some amount of calcium comes into this sodium-calcium exchanger, which is running in reverse mode. So these are all of the way calcium comes into the cell to activate the monofilaments. The monofilaments contract, and then for relaxation, calcium either goes back into sarcoplasmic reticulum through this thing called the circopump, or else it goes out across the cell membrane, either from the action of the sodium-calcium exchanger or this sarcolemmal calcium pump. The sarcolemmal calcium pump is actually very slow. Maybe 1% of calcium goes out that way. Much more of it goes out to this sodium-calcium exchanger. This here breaks down the steps in a little more granularity. Calcium comes in through these voltage-gated channels. It goes into something called a dyadic space. So it's sort of a small area, and then calcium comes out of sarcoplasmic reticulum. Then it generates a calcium transient in the cell, and again, sodium calcium stringer can run in the reverse direction. There's a lot of buffers in the cell, so calcium can bind to component, but there's other buffers, that's just shown here. And then calcium gets pumped back into sarcoplasmic reticulum, and then later in sort of for the turning off the muscle, calcium also gets pumped out across the cell membrane or back into sarcoplasmic reticulum. You can read this the slide here. The interesting thing is when you have about 70% of the calcium is cycled from sarcoplasmic reticulum, and when you get to larger animals, someone gets less. If you're in something like a mouse or a rat, about 95% of the calcium is coming in and out of sarcoplasmic reticulum. Again, it's sort of an adaptation to allow the system to run very quickly. A mouse heart goes at about 600 beats per minute, so you're just really very quickly moving calcium in and out of sarcoplasmic reticulum. This is just showing differences between, say, rat and rabbit. A rat action potential in a cardiac cell looks more like a neuron, very short and quick. A rabbit or a human is much longer, about a third of a second. And basically what this is saying is that during the action potential, calcium is coming in the whole time. And this, unfortunately, is one of the complexities of biology, is that calcium channels both affect the shape of the action potential. They let calcium into the cell to activate the monofilaments. They're also important to load sarcoplasmic reticulum. And probably one of the most important things about hard muscle is that unlike skeletal, that was more or less all or none, meaning every time you activate it, you get more or less the same twitch. When you get to the heart, when you pace it faster, the amount of calcium in each twitch goes up. So if this is a slow pacing rate, 0.5 hertz every other second, now we go up to a faster rate, which is 3 beats per 2 seconds. Then you see that the twitch force goes up. And twitch force goes up mainly as a result of more calcium being released. And then when we slow back down the pacing rate, now the force goes back down. So this is something we know, right? When we get scared, our heart goes faster, and you can feel that there's more intense contraction. So, this is how in the heart you can change the amount of contraction force. You can change the pacing rate. You can also have second messenger hormones will affect the strength of contraction in your heart. So basically, I'm sorry, I'm kind of running a little fast here. Let me go back just a couple slides. This particular spot here, David Yu, who I know you know of, who passed away last year, did a lot of work around these voltage-gated calcium channels. And so this is part of the complexity of this system, is that calcium comes in to this voltage-gated calcium channel. It triggers a secondary release from sarcoplasmic reticulum, but then that calcium either goes out into the cell, but it also can inactivate this channel. So it's like a negative feedback system. So the channel opens, calcium comes in, and then it feeds back on this channel and turns it off. There's also this graded behavior. So the more calcium that comes in, the more calcium that gets released. So that's like a positive feedback system. So some amount of calcium comes in, you even get more release. And then during the course of an action potential, a lot of the calcium later in the action potential just comes in and then gets pumped into sarcoplasmic reticulum. That's shown here. So calcium late in the action potential isn't really activating the monofilament, it's coming in and getting pumped into sarcoplasmic reticulum. I also say here that breaking things into these discrete steps is kind of artificial because it turns out as calcium is coming out of sarcoplasmic reticulum, it's also being pumped in. It's sort of like you would think it's futile. I mean, why do it that way? But essentially, there's no way to so precisely control these mechanisms. So again, this is sort of a textbook view. This is why we build mathematical models, because they let us sort of explore that interactivity and understand it in more detail. Almost there, smooth muscle. So smooth muscle, what is it? It's a muscle that surrounds our hollow organs. That is all the hollow organs except for the heart. So smooth muscle is not striated. One important point about smooth muscle is, though, that it generates about the same amount of force per cross-sectional area as skeletal muscle. This contraction speed is much, much slower. So don't think about smooth muscles being weak, but it just does not contract very quickly. There's two main kinds. There's something called single unit or multi-unit. So here in the single, we call it single unit because you actually have a number of muscle cells, but they're connected together by these gap junctions, so they act more like a single unit. So this would be a nerve coming in, an intrinsic nerve from, say, an autonomic center. It's been activated. So once you activate one of these cells, it's going to pass cell to cell. There's also something called multi-unit, and here, basically the neuron or the nerve is going to terminate on each of these fibers and individually activated. So there's no interconnecting bridges in multi-unit. It's sort of one for one in terms of nerve endings and cells. This is shown a little more schematically here. So here in single unit, so on this side, the cells are connected with gap junctions. So once you excite one, it's going to pass from cell to cell through gap junctions. Over here, in this multi-unit, you see that there's an ending of varicosity or termination on each of these cells. So these more or less get activated individually. One of the most important things here about smooth muscle is that it's activated both by nerves and also circulating hormones. So we kind of sort of know this, that our skeletal muscles pretty much react to input of command signals, whereas our smooth muscle, like our stomach, right, sometimes we get agitated in our stomach churns. So our smooth muscles are activated by circulating hormones. So this is showing the microstructure, and you see many of the same players here, like sarcoplasmic reticulum. Down here you see mitochondria. The important thing you see here is it's not striated. If you look at this level, you can see these small dots and big dots. It turns out there are thick and thin filaments, but they're not arranged in this like sarcomere, almost crystalline structure like it is in striated muscle. So this is showing another diagram. So this would be one smooth muscle cell, and this would be another smooth muscle cell. So if you look inside of each cell, you do see thick and thin filaments in a way. This is a thick filament with myosin, and this is a thin filament with actin. But now they're connected to these dense bodies. And they're sort of woven together in a way that's not as orderly. The sarcomeres, so you have these dense bodies. You also have these intermediate filaments that connect together the dense bodies. So again, this is another cytoskeletal element. And then you have the mechanical junctions that couple cells together so cells don't pull apart and they trap. And then you also have gap junctions for electrical and chemical communication. So we went through this slide already, but this becomes very important in smooth muscle because it's mainly activated through the action of phosphorylation. So let's look at what happens here. So, the big theme around smooth muscle is that you have a number of mechanisms that can activate it. So you can have a receptor-activated calcium channel, so there's something from the outside, a neurotransmitter or hormone. It's going to bind to a channel, and it's going to let calcium in. You can also have, sorry guys. You can also have voltage-gated calcium channels, which let calcium in. You can have, there's another way that calcium comes in through the calcium exchanger. That's the same as the other muscle types. There's neurotransmitters that can, now this is called a G-coupled receptor. And what it does is it turns something called TIP2 into IP3, which is going to cause a secondary release from sarcoplasmic reticulum. So this is somewhat different from the other muscle types. So IP3 is the second messenger. It causes calcium to come out of sarcoplasmic reticulum. So there's different mechanisms which cause calcium to go up, and then similar to the other systems, then calcium has to go down by either pumping it back outside, over here, pumping it back across the cell membrane, or else it gets pumped back into sarcoplasmic reticulum. So now, so how do we actually activate the actinomyosin? So in smooth muscle, it's activated by something called myosin light chain kinase. So as calcium goes up, it binds to calmodulin, and calmodulin will activate myosin light chain kinase. And when myosin light chain kinase activates myosin, it begins to cycle. Sorry, it's going to run over just a minute or two, I hope. And so basically, with this myosin light chain kinase, you go this direction, you activate actinomyosin, you get contraction. So for every kinase, there's a phosphatase. So there's a myosin light chain phosphatase, which takes you back the other direction and turns off the muscle and turns off the actin-myosin interactions. We didn't get to talk about it much in the beginning, but if we look at how myosin is arranged, there's actually 4 subunits. There's 2 heavy units, which give you like the head and the tail. But then there's also these light chains. And these are smaller subunits. There's the essential light chain. This is required for the myosin to interact with actin and generate force. But there's also this regulatory light chain. So in smooth muscle, this regulatory light chain is very important because this is what gets phosphorylated, and this is what allows the myosin to interact with actin. This is just showing the structure of the myosin light chain kinase. So this is some more simplified diagram. So the calcium is coming in either through voltage-gated or ligand-gated channels. You can also get calcium being released from sarcoplasmic reticulum through the action of IP3. This is called IP3-induced calcium release. So now calcium goes up in the cell, it binds to calmodulin. Together those things activate myosin light chain kinase, and that's going to phosphorylate myosin. So here it's like resting and detached. Once you phosphorylate it, it will begin to cycle and cause contraction. And so here's sort of cycling here. And then the interesting thing and the important thing about smooth muscle is when you lower the calcium level, you can stop the actinomyosin from cycling and it will stay attached. This is called a latch bridge. So as we were talking before, you know, in skeletal muscle, when I'm pushing on here, I'm generating force, it's isometric, my muscle isn't contracting, but I'm using ATP, even though I'm doing neural net work. Now, if we think about what's going on in smooth muscles, say around the capillaries, if you want to clamp down on the capillary, what happens is you use the actinomyosin, which will cycle, and you can contract. But at some point, you may just want to hold that force. And so by slowing down the cross-bridge cycling, now essentially we contract when we hold the force, and we stop using ATP. And so that's the critical part about energy usage. Since your smooth muscles, you know, may just sort of be holding something in a particular position for a long time, it makes much more sense to control it this way, where you're actually modulating actin-myosin interactions. So what this is showing is the force-velocity relationship. And so, again, I'm sorry to keep changing different terms, but stress here is force, and this is velocity. And so this would say the fast skeletal muscle would have a curve like this. So it has about the same maximum force, but it goes to a higher velocity. This would be a slow twitch skeletal muscle, and this is smooth muscle, way, way, way down here. So again, it can generate as much force, but it's much slower in the contraction rate. And then this is showing the force-velocity relationship will change as a function of phosphorylation level. So if you go, this is each trace is showing more and more phosphorylation from 10% up to about 60%. So you get the maximum force at a very low level of phosphorylation. Here, the shortening of velocity is a function of phosphorylation. So again, really the key point here is that smooth muscle can first activate itself, the cross-bridge cycle. You can get contraction and force, but then as the calcium level goes down, you can maintain this cross-bridge with something called a lattice bridge. These last two slides, the only point of these were that, you've been in only 3 lectures, and if you were to open up a paper, I think this was published just a couple of years ago, all this stuff should look familiar. This is really kind of the Huxley model here. This is another paper that I just pulled out last year. This has to do with the model of capillaries in the brain, but all of these things now should make sense. So this is just to make you feel good that in 3 lectures you learned a lot. You can open up a journal today and begin to. Okay, so I'll see you in a week. We'll wrap up.